Hello everyone, and welcome back to our channel. Today, we have an exciting topic to discuss: killed vaccines. Vaccines play a crucial role in protecting us from various diseases. So, in this video, we'll dive into the world of killed vaccines, exploring how they are developed, their benefits, and their importance in public health. Let's get started. What are killed vaccines? A killed vaccine, also known as an inactivated vaccine, is a type of vaccine that contains a killed or inactivated form of the disease-causing pathogen. These pathogens can be viruses or bacteria. The purpose of a killed vaccine is to stimulate an immune response without causing the actual disease. What are different methods of inactivation to create killed vaccines? There are several methods of inactivation used to create killed vaccines. Here are some commonly employed techniques. Heat inactivation. This method involves exposing the pathogen to high temperatures to render it inactive. Heat can denature the proteins and genetic material of the pathogen, preventing it from causing disease. However, it is essential to balance the heat exposure to ensure the pathogen is inactivated while preserving its antigenic properties. Chemical inactivation. Chemicals can be used to inactivate the pathogen. Commonly employed chemicals include formaldehyde, beta-propiolactone, and binary ethylenimine. These chemicals modify the proteins and genetic material of the pathogen, preventing replication and disease-causing ability. The concentrations and exposure times of the chemicals need to be carefully controlled to achieve effective inactivation. Radiation inactivation. Ionizing radiation, such as gamma rays or X-rays, can be used to inactivate the pathogen. The radiation damages the genetic material of the pathogen, disrupting its ability to replicate and cause disease. Radiation inactivation is a precise method that requires specialized equipment and protocols. It is important to note that each method of inactivation has its own considerations, including potential effects on the pathogen's antigenicity, the stability of the vaccine, and the safety of the inactivated product. Scientists select the most appropriate inactivation method based on the specific pathogen and the desired characteristics of the vaccine. Thorough testing and quality control are crucial to ensure the vaccine is safe and effective. What is the purpose of inactivated vaccines? The purpose of a killed vaccine, also known as an inactivated vaccine, is to stimulate an immune response in the body without causing the actual disease. It offers protection against specific pathogens by introducing the inactivated or killed form of the pathogen into the body. The main objectives of a killed vaccine are: disease prevention. The primary purpose of a killed vaccine is to prevent the occurrence of a particular infectious disease. By introducing the inactivated pathogen, the vaccine stimulates the immune system to recognize it as a foreign invader. This triggers the production of specific antibodies and the activation of immune cells, preparing the body to mount a defense against the live pathogen if encountered in the future. Immune memory development. Killed vaccines help the immune system develop a memory of the pathogen. Even though the pathogen is inactivated, the immune response generated by the vaccine allows the body to recognize and respond more rapidly and effectively to the live pathogen in case of future exposure. This immune memory provides long-term protection and can prevent severe illness or complications associated with the disease. Public health impact. Killed vaccines have a significant impact on public health. They are crucial tools in preventing the spread of infectious diseases and reducing their burden on society. By vaccinating a large portion of the population, killed vaccines contribute to the concept of herd immunity, where a significant proportion of individuals are immune, protecting even those who are not vaccinated or have weakened immune systems. Safety. Killed vaccines are considered safe because the inactivated pathogen cannot cause the disease it was designed to protect against. This makes them suitable for individuals with weakened immune systems, pregnant women, and those who cannot receive live vaccines due to medical conditions. Global eradication efforts. Killed vaccines have played a vital role in global eradication efforts against certain diseases. For example, the killed vaccine for smallpox was instrumental in eradicating the disease worldwide. Similarly, killed vaccines have contributed to the control and near elimination of other diseases such as polio, hepatitis A, and pertussis. What are the examples of killed vaccines? Certainly, here are some examples of killed vaccines. Polio vaccine. Inactivated polio vaccine, IPV. The IPV is a killed viral vaccine used to protect against polio, a highly infectious viral disease. It contains inactivated poliovirus strains, stimulating the immune system to produce antibodies and provide immunity against polio. Hepatitis A vaccine. The hepatitis A vaccine is a killed viral vaccine that protects against hepatitis A virus, which primarily spreads through contaminated food and water. The vaccine contains inactivated hepatitis A virus particles, triggering an immune response to provide protection against the disease. Whole cell pertussis vaccine. Pertussis, also known as whooping cough, is a bacterial respiratory infection. The whole cell pertussis vaccine contains inactivated Bordetella pertussis bacteria. It is often given in combination with other vaccines in a combination vaccine called DTaP, diphtheria, tetanus, and acellular pertussis. Rabies vaccine. The rabies vaccine is a killed viral vaccine used to prevent rabies, a viral disease transmitted through the bite of infected animals. The vaccine contains inactivated rabies virus particles and is commonly administered after a potential exposure to prevent the development of rabies. Inactivated influenza vaccine. The inactivated influenza vaccine, commonly known as the flu shot, is a killed vaccine used to protect against influenza viruses. It contains inactivated strains of the influenza virus, covering the specific strains predicted to be circulating in a given flu season. Japanese encephalitis vaccine. Japanese encephalitis is a mosquito-borne viral disease prevalent in certain parts of Asia. The Japanese encephalitis vaccine is a killed viral vaccine containing inactivated Japanese encephalitis virus strains. It provides immunity against the disease and is recommended for individuals traveling to or residing in high-risk areas. Conclusion. Killed vaccines have made a tremendous impact on public health. They have helped to eradicate diseases like smallpox and have greatly reduced the incidence of others. By vaccinating a large portion of the population, killed vaccines contribute to herd immunity, protecting even those who are unable to receive vaccinations. They are a vital tool in preventing the spread of infectious diseases and saving countless lives. That's all for today's video. If you found this video helpful, don't forget to give it a thumbs up and share it with others. Thank you for watching, and we'll see you in the next video. Stay safe and stay healthy.